Hello, students. Today, we're going to explore some really fascinating methods of asexual reproduction that are used by simpler organisms. It's quite an interesting topic. So, let's have a quick look at the topics we will be covering today. First, a little introduction, then we'll get into the three main methods: fragmentation, regeneration, and budding. And at the end, we will do a quick summary to make sure all the concepts are perfectly clear for you.
All right, first, let us recall the basics of asexual reproduction before we move on to the different methods. Now, the most important point for you to remember is that asexual reproduction involves only one parent. This is very common in organisms that have a simple body design.
Okay, let's dive into our first method. This one is called fragmentation. As the name suggests, it has something to do with breaking apart. So please note the definition carefully. Fragmentation is a type of asexual reproduction, where an organism, usually one with a simple body, breaks up into smaller pieces or fragments when it becomes mature. A classic example that we study for this is Spirogyra. It follows these very simple steps to reproduce. First, the organism matures, then its body breaks up into smaller pieces, and finally, each of those fragments grows into a new, complete individual. Simple, isn't it? Now, this leads to a very, very important question. Why can't more advanced animals like you and me reproduce this way? Well, the crucial point is that our bodies are far too organized and complex. You see, we are not just a collection of cells. Our specialized cells form tissues, tissues form organs, and all of these are at very definite positions. So a simple cell-by-cell division like in fragmentation would just be impractical. It cannot work.
Okay, let's move on to our next method, which is regeneration. Now, please pay attention here because we need to see how it is different from fragmentation. This is an area where students often get confused.
Now, what's really interesting about this slide is it highlights the main confusion. You see, fragmentation is a method of reproduction. Its purpose is to create new individuals, but regeneration is an ability an organism has to regrow lost body parts. They are not the same thing.
So, in regeneration, if an organism, like, say, a planaria or a hydra, is somehow cut or broken up, each of those pieces has the amazing ability to grow into a complete new organism.
You see, this process is not at all random. It is a highly organized sequence, and it all happens because of specialized cells. These cells multiply very rapidly. They form a large mass of cells, and then these cells change to form different cell types and tissues. This whole organized sequence is called development.
And this quote you see on the screen, it brilliantly illustrates the key difference for your exams. Organisms don't normally depend on being cut up to be able to reproduce. That's why regeneration is more of a survival mechanism than a primary method of reproduction. All right, our third and final method for today is budding. This one is all about growing a new individual. So in this process, a new individual grows out from the parent's body. This outgrowth, which we call a bud, develops because of repeated cell division at one specific site on the parent. Hydra provides a perfect example for us to understand budding. It uses its regenerative cells to form a small bud. This bud keeps developing into a tiny individual while it's still attached to the parent. Then, when it's fully mature, it just detaches, and it becomes a new independent individual. And it is important for you to note that budding is not limited to multicellular organisms. It also happens in single-celled ones, and a very common example of that is yeast. Now, let us summarize everything in a single table. This will make it very, very easy for you to remember and revise for your exams. This table clearly lays out the mechanism and the key examples for each method. So for fragmentation, the body breaks into pieces. For regeneration, it is the regrowth of parts from specialized cells. And for budding, an outgrowth or bud forms and detaches. This is a very important slide for your revision. So, what is the crucial takeaway from all this? Well, even though these methods are simple, they show the incredible potential of cells to grow and develop into a complete being. And this is a fundamental concept in all of biology. Thank you.